So there is a book about the war in the Chinese history written by Sun Tzu, and he said that the super art of the war is to subdue the enemy before fighting. So actually, maybe we need to focus on the T cells, right? And of course, we know the importance of the T cells compared to antibodies, so we go through this quickly. And actually, in my group, actually we have done a lot of work on the MHC moleculars, especially in the different HLA alleles of humans. And also we focus on different viruses, especially including the coronaviruses and also the influenza. And also we focus on different species, especially the mammals. Together we have already published about more than 100 MHC molecular structures, I think, and hope in the future we can have the chance to collaborate with ProImmu. And also this is the 12 animals, right, in China. Actually, almost all the animals we have the MHC moleculars already. Of course, we do not have the structure of dragon yet, but we have determined the structure of bat. But actually, Professor George Gao do not think bat can, representative dragons. So maybe in future we can done some work of other animals. So actually, my work today to talk is about the influenza and COVID-19. So actually, in the beginning of the COVID-19, we have determined the immune memory of the after the COVID-19 about 6 months, and also 12 months recovery, and we determined the antibody and also T cell responses, and we found that especially the T cell responses, if we use the peptide cultured cells in vitro, and we can determine over 90% of the recovered patient have the memory T cell responses, and also the T cell responses have the collaborating with the severity of the infection. And also we can determine the specific T cells, of course, using the multiple of the MHC moleculars. That is the memory about the T cell responses. And also we have some work about the cross-reactivities between different variants, especially for H1N1 and H5N1 influenza viruses. And of course, we know there is no cross-reactivity between the antibodies. But actually, there was broadly cross-reactivity between T cells, especially between H1N1 and H5N1. And we have also done some cross-protection in mice, and we found the H1N1 pre-infection can contribute to the protection against the H5N1. And also for the recent H5N1 in North America, and actually we have go through all the potential cross-reactivity between H1N1 and H5N1, and we found that, of course, there was a level of cross-reactivity to H5N1 in the recovered patient of the H1N1. And actually, we have done some specific tests in HLA-11 and HLA-33, and actually they have different cross-reactivity to H5N1 levels. And also we have done some cross-reactivity between vaccinian viruses and also the monkeypox viruses. And because in China we have injected the vaccinium Tiantan strain about 40 years ago, and actually we can find the cross-reactivity we can find the memory antibody responses, but actually we have some cross reactivity of the antibody against monkeypox for the antibody. And actually we found also a broad cross-reactivity to monkeypox in the population who are injected by the the Tiantan strain of the vaccinia virus. And also for the cross-reactivity between the SARS-CoV-2 and also other coronaviruses, we found this is very common between different coronaviruses. And actually we found in the very beginning of the SARS-CoV-2, the common cold coronavirus can play a pre-existing reactivity to COVID-19. But actually nowadays, nearly everybody have the basic T-cell responses to COVID-19. So actually nowadays, I think we have a reverse pre-existing immunity to common coronaviruses by the COVID-19. So actually we determined the cross-reactivity between SARS-CoV-2 viruses and also the SARS coronaviruses. They have a broad cross-reactivity between each other. And also, although there were cross T-cell responses, but actually we have to admit that the T-cell response escaping also exist in different viruses, especially in the influenza and also the COVID-19. So this is similar as the red queen hypothesis. So let's see the JN1, JN.1 or 2.86 variant of COVID-19, and we found that most of the variant mutation sites are located in the different T-cell epitopes, especially this kind of overlapping T-cell epitope. We can call it hotspot of T-cell epitopes. And we found that especially this peptide, it is a HLA-A24 restriction, and actually we found from the very beginning, the Delta, they have already emerged a mutation, but actually until the JN.1, they have a series of mutation on this peptide, and we evaluated the spectrum of the escaping, and we found that the Delta and the BA.5.2 and also the JN.1 have a very high T-cell escaping. It is for the the arginine and also the serine in the 455 mutation. And we determine all the the structures of the peptide complex to the HLA-A24, and we explained why these two mutations can escape from the T-cell recognition, because the electricity of the surface of the peptide have changed. Interestingly, we have also determined another site. It is in the N-protein. As we know, the N-protein in SARS-CoV-2 is very conserved, and we can see the even for all the subaqueous viruses, and also the viruses, SARS-CoV-2 viruses before the JN.1, they are all conserved for this site. It is 229. It is a Q residue. But actually, only for the JN.1 and also the BA.2.86, it have a mutation from Q to K. So actually, we found that this mutation located in a series of HLA-A2 restricted peptide, and we evaluated the immune escaping and all the is HLA-A2 peptide have a mutation, immune escaping, and also we determine the structure, and this residue are located in the surface of the peptide and can be recognized by the T cell receptors. So that is why it can escape from the mutation recognition. And actually, we have some following study about this site. That paper is about gene.5, but actually we also determine other, mutations after that, like KP.3. And actually, generally, we can find, the peptide pool have a cross-reactivity between each other. And actually, we also find some public TCR quite similar as Professor Dong Tao's work, and we found this individual can cross-reactive, nearly all the peptide, including the mutations. And actually, we found another very important residue is 346, and actually, they also have, site is very important because it is recognized by the neutralization antibody and also the ACE2 binding, so it is have a very high mutation rate. But actually, we found all the the mutation cannot escape for the T cell recognition. Of course, another peptide we can also got the same conclusion. So actually, for our take-home message is that we can see the T cell immunity can sustain at least years, and also we can see a universal cross-reactivity between the different virus variants, and also although the cross-reactivity also exists, but actually the T cell immune escape also exists, but actually T cell immunity is one of the driving force of the virus mutation. And this is my another duty is introduce the journal H-Life, and welcome you to submit your papers and perspectives. Some other and also another paper, another journal is Biosafety and Health, and actually this is published in China CDC, and welcome to publish your work. And this year is November 1st is the 8th World Flu Day, and we will have this symposium in Wuhan, China. Okay, the last slide is is my new group in Guangzhou lab. If you have a student or postdoc, welcome to recommend it. Thank you very much.